talk about the first set of cytoskeletal filaments. In this case, it is the actin cytoskeleton. And so here you can see a EM micrograph of what the actin filament looks like. It actually looks like the cartoon drawn here too. It has a little twist to it. You can see the little bit of subunits, and so that's remarkably close. You can also see what it looks like with immunofluorescence. And so this is the actin fibers that are stretching throughout the cell. And so you can see that these fibers are actually quite long. They encompass almost the whole stretch of the cell at times. And so you have to really form these very, very large multi-subunit proteins to generate that. Okay? So this is the last reminder of the evaluations. You go to this link. It's also, someone asked a question on the discussion board. We put the link there as well. Again, it's part of your grade, and it's due before 1 p.m. on Friday. And so please, please, please do that. About 60% of you have taken it. All right. So in this lecture, we're going to talk about actin and actin-binding proteins. We'll talk about how they assemble and disassemble. We'll be discussing the myosin II cycle as well, a motor protein that walks along actin.
Look over here, guys.
As with most families, the McKittrick boys received many of the traits of their parents, their smiles, their hair color, their eyes. But Todd and Lane McKittrick also passed on a rare genetic disorder of which they had no idea they each carried.
Neither of our families throughout the ancestry has ever been diagnosed with Usher syndrome. So it was a 1 in a million shot for us to get it together.
2 of their 4 boys have Usher syndrome, marked first with deafness and then eventual blindness.
Abracadabra!
Even they didn't know their firstborn, Connor, had Usher syndrome right away. They knew he was born deaf, so early on he received cochlear implants. It wasn't until he was 5, when his eyesight began to fail, that the diagnosis came. When their youngest, Dalton, was born deaf, they knew that lightning had struck their family twice.
Alright, Dalton. Ooh, look at this place.
I'm trying to freeze you.
Freeze ray!
Because we, for many years, kind of felt sorry. You know, it was like, oh my gosh, how can this be happening to us? And Connor's taught us that, you know what, there's nothing wrong with me. It's hard. I go through lots of hard things during the day, and I wish I could change it, but I'm here for a reason, and I'm here to help others, and so I want to help my little brothers.
I'll try to tackle him.
I'm not going to feel sorry for me. I'm going to help him because I've already been through it, and so he sort of accepted it. There's many times when we still don't accept it, but it's just part of our daily life.
Revenge!
Oh. It's an abrupt stop. So that's Connor and Dalton, and they have Usher syndrome. And so this is what Usher syndrome is. It's genetic. And so when the reporter said lightning struck twice, that's actually not the case, right? So we know that there is a 25% chance with genetics that 2 recessive-bearing individuals will have a homozygous recessive child. And so here are the different types of Usher syndrome, Usher type 1, 2, and 3. And so you could have deafness at birth and visual impairment in the childhood. You could have type 3, worsening of hearing loss with late onset visual impairment. And so obviously type 3 would be most advantageous to have if you had this, not type 1. And essentially what happens is you have these cells in your ear, for instance, that will hear these action potentials. And so they, during resting, will have a certain amount of action potentials. When sound, the mechanical force of sound ends up brushing these little stereocilia, what happens is you get stimulation, and so you get more action potentials occurring, and this is what allows you to hear. And so when this process is disrupted, you can go deaf. You also have the same process for your eyesight. And so that's why you would go blind eventually, is if you no longer had light actually hitting these little filaments, then you would no longer be able to see. So that brings us to the first clicker question, which is, Circe is interested in stereocilia formation as her children are genetically predisposed to recessive genes. If actin makes up the rigid structure of stereocilia, how does the structure persist given the dynamic nature of actin filaments? All right, so what you'll see is about half of you said B, which is actin treadmilling. About 25% of you said A, high monomeric actin concentration preferentially incorporates at the barbed end of the filament. And so what is the correct answer? It's actually A, actin treadmilling. And so why is B, actin treadmilling. Clearly I'm dyslexic. Okay, so why is B correct? And so here are stereocilia, the same little things that you saw in the cartoon where sound vibrations would deform them. Here is what they look like. So actin is labeled in green, and so this is monomeric actin. And then phalloidin is labeled in red, and so phalloidin is actually binding to preformed actin filaments. And so what you'll see is that normally you have all of these preformed actin filaments, and that when you add in actin monomers that have this GFP, you can see that they preferentially incorporate at the tips of these stereocilia, and then over time they start to slowly take over that stereocilia, and by 48 hours you've essentially taken over the whole stereocilia. So this is an actin-based structure, and it incorporates monomeric actin at the tip. But then you can see it's a relatively slow process where it takes about 48 hours for the whole thing to actually occur. Okay? And so it's a very slow process in this instance. Now, this is what it actually looks like in a cartoon format. You have actin monomers incorporating at the tip. And then as this treadmills, you can see that the actin monomers, the GFP actin monomers, will slowly march down here. Because what's happening is you get preferential incorporation at the tip, and then you get depolymerization, disassembly at the tail end. Okay? And so eventually, over 48 hours, this will completely take over the whole filament. Now, the reason why it's so stable is you have all these other proteins bound. And so in this case, this is this ESPin GFP. This is a actin-binding protein. It binds to filaments, and it helps stabilize them. And the stabilization makes this process incredibly slow. Without that stabilization, actin GFP would probably take over this whole filament within seconds, right? And so that kind of gives you a bit of information as to how much these actin-binding proteins can influence the process of actin incorporation and disassembly. Okay? And so here's what the stereocilia look like. And you have all of these proteins that actually interact with the tip and connect these stereocilia together. And then this whole process actually influences the ability of these stereocilia to signal. And it's these processes that are disrupted in Usher syndrome, these proteins. And so we'll get into more of Usher syndrome a little bit later, but I want to talk a bit more about treadmilling. And so this is essentially a picture taken directly from your book. And what you can see is this is an actin filament initially forming. And so what you can see is that there's a T form and a D form of actin. And the T form is then preferentially added at the very beginning. And then as the filament is actively growing, the T form is then preferentially added at the plus end and not the minus end. And so this is what it looks like. This is the T form of actin, and this is the D form of actin. So what you can see here is this is subunit concentration, and this is the elongation rate. And so we can see that the elongation rate is much quicker with the T form of actin than the D form of actin. And then this is the treadmilling range where the critical concentration is between the critical concentration of the T form versus the D form. Okay? So what is the T form of actin? What's the D form? Does anyone know? So it's bound to ATP and ADP. Yes, not GTP, different energy. And so in this case, actin, the subunit, the T form is the ATP bound form, whereas the D form is ADP bound. Okay? Let's see here. And so what you can see is that addition of monomers requires you to be in your ATP bound form, not your ADP bound form, right? You only add the squares, not the circles. That being said, you can see that there is preferential, that you can add at both ends. Initially, when this first starts, you can add at both ends. Over time, oftentimes you get preferential addition at one end versus the other because of the actin subunit concentration. But essentially, here are the rates, right? And so the rate at adding to the plus end is greater again than adding to the minus end. But again, they have to be the T form, ATP bound. Okay? All right, so that brings us to this next question. Circe monitors stereocilia with actin GFP. She perturbs stereocilia by adding cytoclasin B, which caps the barbed end. What happens to actin and to stereocilia? All right, you can see that 50% of you say B, which is stereocilia shrink. And then roughly 15 to 20% of you say C or D, actin GFP only incorporates at the plus end, or actin GFP does not depolymerize at the minus end. And so what is the correct answer? And you'll see it's B, stereocilia shrink. And so if we look at what is actually happening when you're capping different ends. And so we have the plus end and the minus end. The plus end is also called the barbed end. And so when you cap the plus end, where you get preferential addition because of the rates, this completely changes the rates at which things can add. And so here is, again, the critical concentration. So the green line represents filament growth essentially at both ends. The red line represents filament growth at only the minus end. And so yes, filaments will grow at the minus end, but it is far, far slower. And if you're actually in the treadmilling range, you actually get shrinkage of a preformed monomer instead of slow growing. And so what does that actually look like? So here is cytochalasin, and here is the control. And so this is stereocilia after 24 hours, and you can see the actin GFP incorporation, and this slowly marching down the filament. And then here's cytocalasin treatment. And so they added the actin GFP at the same time as cytocalasin. And so you have some of the actin GFP actually incorporating at the tips before cytocalasin blocked it, right? But you don't see it march down. And so if you look at stereocilial length over time, you can actually see that it shrinks. That cytocalasin, when you block the barbed end, the plus end, you eventually get shrinkage when they're at a treadmilling concentration. Okay? What would most likely happen to stereocilia if Circe adds phalloidin, a compound which binds to and stabilizes the actin filament? Okay, what you'll see by your responses is a little over 40% of you say stereocilia continue to treadmill, E, and then about 30% of you say stereocilia grow. Now, what I'd like for you to do is actually talk to your neighbor, discuss this question, and then I want you to click in again.